Hello again, everyone. Here's the final lecture for week 5. This lecture is about bioenergetics and oxidative metabolism. And all about it, this is all talking about how we make ATP for energy. Why do we care about this? Well, oxidation of metabolic fuels is essential for life. So fuels such as carbohydrates and lipids are broken down. I talked about how lipids are broken down. You've heard about how carbohydrates are broken down from the TCA cycle. They're all metabolized to carbon dioxide and water, basically leading to ATP, or the central metabolic currency, adenosine triphosphate. The majority of the metabolic energy is produced by these oxidation reduction reactions that I'm going to talk about today, and this occurs in the mitochondria. We've talked about the mitochondria before, but obviously this is important for cellular energy. It's also important for signaling, cellular differentiation, cell death, the cell cycle, and cell growth. There's the outer membrane and the inner membrane. A lot of the action for oxidative phosphorylation takes place in the inner membrane, and that's what we're going to be talking about today. So there's 3 main topics, with the majority of the lecture being on oxidative phosphorylation. But the first thing we're going to talk about is oxidation as a source of energy. The caloric value of fuels or foods are calculated by complete burning or complete oxidation. So the more reduced your fuel is, the higher calorie per gram of your fuel is going to be. So for example, we've talked about in the past, fats are more reduced than carbohydrates and proteins, so they have a higher kcal per gram of molecule. You can also see alcohol is even higher calorie density than carbohydrates. So when we talk about energy, 40% of food energy is conserved as ATP, and approximately 60% is released as heat. Almost all the reactions in the body are exothermic, meaning they release heat. If we talk about the sum of all reactions at rest, this is what our basal metabolic rate is. You can actually measure this directly through measuring of total heat released over time, or indirectly by the amount of O2 consumed. And we'll talk about that, monitoring partial pressure of O2 later in the lecture here. The heat production by the mitochondria accounts for most of the basal metabolic rate. How do we conserve energy in the form of ATP? Well, it's due to this process of oxidative phosphorylation that we're going to talk about. There are 2 stages. First, 2 stages of fuel oxidation. The first we've already talked about, I've already talked about, and when other lecturers have talked about the oxidation of fuels, I talked about the oxidation of lipids. During that process, you produce reduced nucleotide coenzymes. What we're going to be talking about today is the use of the free energy from the oxidation of those reduced coenzymes to produce ATP. So in a very simplistic form, you have your fuel, you produce reduced coenzymes from that fuel, whether that fuel is a lipid or a carbohydrate or protein. And then in the second stage, you use the free energy from the oxidation of these reduced coenzymes to produce ATP, and we'll talk about exactly how that works. Before we do that, let's take a step back and talk about the concept of free energy. Free energy, or Gibbs free energy, is the maximum amount of energy that can be obtained from a reaction at a constant temperature and pressure. The units for that are kcals per mole or kilojoules per mole. It's mathematically defined pretty simply. It's defined as the free energy content of the final state minus the free energy of the initial state. So if we have a reactant A that forms a product B, the free energy change in this reaction, or delta G, can be defined by the following. Delta G equals the Gibbs free energy of B minus the Gibbs free energy of A. So this is very simple. and for all biological reactions, the if we go from A to B instead of B to A, it's simply the opposite value of the Gibbs free energy, because you'd have GA minus GB. Pretty simple. If a concentration of B is greater than A at equilibrium, so if we go all the way to equilibrium, we have higher concentration of B than A, then the conversion from A to B is favorable and you have a negative delta G. And then the exact opposite is true if the concentration of A is greater than B at equilibrium, the reaction is unfavorable and you have a positive delta G. So for a reaction to be favorable, you want a negative delta G. You sometimes see this written with delta G, not with the degree symbol or the accent symbol. This means it's at pH 7. This represents the biological delta G's. The Gibbs free energy can actually be calculated by this equation here, where it's negative RT natural log of the K equilibrium. So where T is the absolute temperature, R is the gas constant, which is shown here, and the equilibrium constant, the natural log of the equilibrium constant for the reaction at pH 7. So again, we don't need to do calculations on the exam, but this just introduces you how it could be calculated. So for example, the hydrolysis reaction of glucose 6-phosphate, glucose 6-phosphate plus water yields glucose plus phosphate. Excuse me, this has a negative delta G value, so the reaction is favorable. Living systems transfer energy from molecule to molecule without losing it to heat, and some of this energy is conserved in chemical form, and about half of the energy from the oxidation of fuels is conserved in the form of ATP. And again, we're going to talk about exactly how that works. The ATP is important because the hydrolysis of the two anhydride linkages in ATP, these right here, yield highly negative free energy. Those reactions have delta G's that are highly negative to form, and then if you cleave one phosphate, you get ADP. You cleave two phosphates, you get AMP. So these are high energy bonds, and when you cleave them, you can couple those reactions to drive other reactions that may need to go in unfavorable directions. So here's an example of that. If we look at the synthesis of glucose 6-phosphate from glucose, we need to go glucose plus a phosphate to yield glucose 6-phosphate. But if you just look at that in a vacuum, it has a positive delta G, so this reaction does not want to go forward. However, if you couple it with ATP hydrolysis, which has a negative delta G, you can combine those two to have the net reaction and overall negative delta G to drive this reaction forward. As a little bit of an aside here, the metabolic function of ATP requires magnesium, and so ATP forms a complex with magnesium, and it's actually this complex that's required for all reactions that ATP is involved in. So if you have magnesium deficiency, it can impair virtually all metabolism, because basically you can't use ATP. What happens when you use a lot of ATP? So when you exercise, you're basically consuming ATP by doing a lot of reactions in your body. You can accumulate adenosine monophosphate, AMP. So this AMP activated protein kinase is actually a fuel sensor. It helps initiate the production of new mitochondria and electron transport components to help you metabolize fuels faster. That's the introduction to oxidation as a source of energy and the constant of free energy. So the remaining slides are going to be devoted to oxidative phosphorylation. So again, the main function of the mitochondria is to oxidize metabolic fuels and conserve energy in the form of ATP. Notably, most mitochondrial proteins are encoded in the nuclear genome and transported to the mitochondria. The mitogenome itself actually encodes 13 subunits of proton pumps and ATP synthase, but a lot of these are encoded on nuclear genomes and they have to be transported to our mitochondria. I showed the picture previously of the outer and inner membrane. The outer membrane of the mitochondria is very permeable to ions and many proteins, but the inner membrane is much more controlled. It's impermeable to most ions and small molecules such as ATP, protons, etc. And so this is how we can form gradients across this inner membrane to drive ATP synthesis. So the inner membrane is where all the action happens. It contains components of oxidative phosphorylation, which again, just to strictly define this, the oxidative phosphorylation is the process of coupling oxidation of these reduced nucleotide coenzymes to the synthesis of ATP. The main coenzymes are NAD+, FAD, and FMD. The oxidation of these reduced nucleotides, so for example, going from NADH to NAD+, produces a large amount of free energy that can be conserved in ATP via this electron transport system that we're going to talk about. Let's talk about these coenzymes a little bit. So here's NADH, FMNH2. If you look at the structures of these, again, the structures aren't terribly important, but if you pay attention to the hydrogen atoms here, these are the reduced forms. What happens during oxidative phosphorylation is these coenzymes get oxidized, and these reactions have a large delta G associated with them, helping to drive hydrogen atoms across a gradient, which enables ATP synthesis. So here's what the whole thing looks like. So we're going to step through this. The electron transport system is known as the electron transport chain or the respiratory chain. Again, this is located in the inner membrane of the mitochondria. So just to orient you on all these slides, this is inside the mitochondria, and this is the inner membrane space, and then the outer membrane would be here if we were showing it. But here's the direction of the electron flow down the electron transport chain. The whole system consists of large protein complexes 1, 2, 3, 4, and 5, with 5 being ATP synthase, as well as some small components of ubiquinone and cytochrome C. Each step along the chain here involves a redox reaction. The electrons are conducted through the system from reduced nucleotide coenzymes to oxygen by flavoproteins. Free energy changes drive the transport of protons from the matrix into the intermembrane space. So you're basically creating this hydrogen gradient to where you have much more out here in the intermembrane space compared to the matrix. And these are primarily driven by 3 proton pumps that are part of complex 1, complex 2, and that should say 3 and 4. So the 3 that are circled in red here. If the flavoproteins that are in the electron transport chain are what helps conduct electrons through the system. There are 4 major types. Complex 1 contains FMN, 2, 3, and 4 contain FAD. For each pair of electrons transported through these flavoproteins, protons are pumped for the synthesis of ATP. Here's the overall reaction shown here. Now we can measure the electron transport chain by using something called the phosphate-oxygen ratio. What this is a measure of the high energy phosphates, basically the amount of ATP that's synthesized per atom of oxygen consumed or per mole of water produced. So here's what a graph of that looks like over time. So you can look at the PO2 upon introduction of ATP into the mitochondria. So you add ATP and that VO2 level goes down, and ultimately that ADP is exhausted. So generally, as we talked about, this oxidation and the phosphorylation are highly coupled. So you get substrates that are oxidized, electrons transported, and then oxygen is consumed only when ATP is produced. And we'll talk about a couple times when that coupling can become uncoupled. But let's talk about the large protein complexes that are involved in this chain, in this electron transport chain. There's 5 of them, as well as the small components ubiquinone and cytochrome C. Many of these contain these iron-sulfur complexes, and this is where all the action is occurring for the electron transport. Complex 1 is also known as NADHQ, the reductase complex. Complex 2 is known as succinate dehydrogenase complex. It's actually a separate starting point to the electron transport chain, and it's not part of the NADH normal pathway. Complex 3 is cytochrome C reductase, complex 4 cytochrome C oxidase, and complex 5 is the ATP synthase complex that's actually taking ADP and phosphorylating it to form ATP. So let's just walk down the chain through each of these complexes. Complex 1, or NADHQ reductase complex. What happens here is NADH binds to an FMN and is reoxidized to NAD+, transferring 2 electrons. Electrons are transferred via a series of iron-sulfur clusters and finally to coenzyme Q10, otherwise known as ubiquinone. Then what happens is you get electron flow changes. Electron flow changes the redox state of the protein, altering the pKa values of ionizable side chains, and ultimately causes 4 hydrogen ions to be pumped out of the mitochondria matrix. And then ubiquinone CoQ accepts 2 electrons. Complex 1 has some clinical importance to it. Obviously the whole chain does, but there's a syndrome called Leber's hereditary optic neuropathy. Basically, you get a single nucleotide change in the mitochondrial DNA that mutates an amino acid in complex 1 from an arginine to a histidine. And it can really, it affects the eye. If normal vision obviously is like this, and the vision with this syndrome gets very blurry, and especially in the middle of the vision. There are complex 1 inhibitors. Rotenone is one. There's also 1-methylphenylpyridine. This retinone can actually produce drug-induced Parkinson's syndrome. It's a clinical syndrome that's characterized by tremor, rigidity, and it's found in Parkinson's disease. And so you can use these partial oxygen pressure graphs. For example, if you look at the uptake of oxygen is stopped when you add retinone. And so they use these graphs as to look for inhibitors, for example. Coenzyme Q is the next stage in the process. It's also known as ubiquinone. It's actually a small lipid-soluble compound. Here's a structure of it. You can see it has an isopenoid backbone. It accepts electrons from complex 1 as well as other flavoproteins and then transfers them to complex 3. You can have deficiencies in coenzyme Q. For example, here's a clinical presentation of a 4-year-old boy that presented with seizures and progressive muscle weakness. He had accumulation of lactate, product of anaerobic metabolism of glucose. He also, this suggests a defect in something that's going on with oxidative metabolism in the mitochondria. They looked at activities of complex 1, 2, and 3 and were normal, but 1 and 3 and 2 and 3 were low. And basically it was because of this transfer of electrons through the coenzyme Q. So they treated him with coenzyme Q, which improved some of his muscle weakness. Complex 3 is important because when these electrons are transferring, complex 3 is hindered either through point mutations, or you can have inhibitors such as antimycin A. A complex 3 may leak electrons to oxygen, yielding the formation of superoxide. And superoxide is very toxic. It leads to a number of diseases. It can also lead to premature aging. And again, you can use these PO2 graphs and see the slowing down of the uptake of oxygen when you add a complex 3 inhibitor in this case. I mentioned cytochromes, and cytochromes are important for this lecture because they complex 3 and 4 act on cytochromes. Cytochromes are actually proteins that are found in the mitochondria and the endoplasmic reticulum. They contain these large heme groups. For example, here's the heme group of cytochrome C. Here's the heme group of cytochrome A. In these cytochromes, the heme iron is reversibly reduced and oxidized between the Fe2+ and Fe3+ states as electrons are shuttled through from one protein to another. So this is part of the iron sulfur clusters I was telling you about that help shuttle electrons through the system. Cytochrome C specifically is a small heme protein that's loosely bound to the outer surface of the inner membrane of the mitochondria. It contains heme C, which I just mentioned on the previous slide. Heme C is actually covalently bound to the protein through cysteine residues. And again, this is where the iron sulfur cluster is, to shuttle electrons from complex 3 to complex 4. We're moving on to complex 4, which is also known as cytochrome C oxidase. It oxidizes cytochrome C and conducts electrons through the cytochromes A and A3, reducing oxygen to water in a 4-electron transfer reaction. The 4 hydrogens are then pumped across the membrane. This series of hydrogen pump step creates a gradient, and then this gradient is used to make ATP, as we'll talk about here in a minute. Before we get there, I want to mention that copper is a common component of this and other oxidized enzymes. So if you have a copper deficiency, and we just get copper from trace amounts from the food we eat, this deficiency is rare in adults, but in premature infants can have low stores of copper and may suffer from deficiency. So this can lead to anemia and cardiomyopathy, basically because you're failing to synthesize adequate amounts of cytochrome C oxidase and other enzymes. So complex 4 is important, just like the other complexes. The failure to synthesize adequate amounts of cytochrome C oxidase and other enzymes can lead to, again, cardiomyopathy, Leigh's disease, which is due to mutations in the mitochondrial DNA, causing degradation of motor skills and eventually lead to death. Okay, we're on to the final complex, complex 4, otherwise known as ATP synthase. So along this electron transport train, we've been building up concentration gradients. These concentration gradients are a key component of the biological world. We talked about concentration gradients last time, driving active transport. The potential energy from those gradients is used to perform work, in this case to make ATP. So the use of a hydrogen gradient occurs in the mitochondria when this ATP is synthesized. ATP synthase is a large complex of membrane-bound proteins. You can think about ATP synthase as a motor generator complex, where F0 is the motor and then F1 is the generator component. The F0 sits in the membrane, whereas F1 is in the matrix component. The F1 actually consists of 3 alpha-beta dimers. So the catalytic activity resides on the beta subunit. In each 120-degree rotation of the gamma subunit introduces a conformational change in the alpha-beta dimeric subunits so that the nucleotide binding sites alter between 3 states. The first one, it binds ADP plus phosphate. Second one, it synthesizes ATP. And the third one, it releases ATP. So each turn produces 3 ATP molecules. So obviously complex 5 is important. I don't think I need to say this, but the complete loss of ATP synthase is not compatible with life. But if you just have deficiencies in the complex, it can lead to myopathy, cardiomyopathy, seizures, lactic acidosis, brainstem degeneration, can lead to coma. So obviously this is a very important enzyme for our bodies. One notable inhibitor of ATP synthase is oligomycin. You can see again on this PO2 graph, when you add oligomycin, you get reduced oxidation, so it inhibits the respiration. It actually does it by inhibiting this proton channel of ATP synthase. There can be some uncoupling, as I mentioned previously. So when we talk about respiratory control, the mitochondria can become partially uncoupled or leaky. It's called uncoupling because the oxidation proceeds without production of ATP, and so the PO ratio declines under these conditions. There are some compounds that can act as uncouplers. Essentially what happens here is you get transport of hydrogens back into the mitochondrion. So it dissipates this gradient that's being built up to produce ATP synthase, and it can stimulate heat generation. These protein uncouplers are endogenous and can be regulated by hormones. There are also, so here's an example of a protein uncoupler, or I guess a diagram I should say of a protein uncoupler. There are some small molecule uncouplers, sorts such as DNP, which is shown here. They are exogenous hydrophobic compounds. Some of them are weak acids, or some could be weak bases. They have pKa's close to about 7. So it's just one example shown here. So here again is just a repeat of the slide I showed earlier in the lecture, but this really sums it all up. Really, you're building this hydrogen gradient down the electron transport chain that is ultimately coupled to the synthesis of ATP. So I know I had a lot of details, but the important things are to understand what the major 5 complexes do and the overall chain, the overall electron transport chain to build up this gradient for ATP synthase. I've talked about inhibitors of some of these complexes. One global inhibitor, or 2 global inhibitors, are cyanide and carbon monoxide. They're actually mitochondria poisons. They do this by binding to hemoglobin and inhibiting oxygen transport, electron transport, and ultimately the production of ATP. So what happens is cells can respond to this poisoning by switching to anaerobic metabolism, but this results in lactic acidosis and can ultimately lead to death. So on that sobering note, I'll leave you guys with the study guide here for Chapter 9. Hopefully hits on the high points for you. But reach out to me with any questions you may have, and look forward to seeing you next time.